Good morning, my dear students. Welcome to my classes. In this session, we are going to discuss about one of the important and most advanced vaccine called as self-amplification, amplifying RNA vaccine. Actually, mRNA vaccines, so far the most advanced vaccines than the mRNA vaccines, these self-amplifying RNA vaccines are much powerful vaccines. So these self-amplifying RNA vaccines, how they work and how these self-amplifying RNA vaccines are being made and how these saRNA vaccines are completely different to that of mRNA vaccines. Don't think that self-amplifying RNA vaccines are the advanced version of mRNA vaccines. Not at all, right? Self-amplifying RNA vaccines are completely based on a new technological platform. So as self-amplifying RNA vaccines are made with completely new technological platform, these are somewhat they have as they're made from a new technological platform, they have tremendous advantages over other vaccines like mRNA vaccine. So first one is what is saRNA, self-amplifying RNA, and how these self-amplifying RNAs are going to be useful and how they are made. Why? What is the context? Because in the last week in Japan, self-amplifying RNA vaccines have been made by the Japanese government authorities has given approval to the saRNA, self-amplifying RNA vaccines. So in the context, we can say self-amplifying RNA vaccines, we'll be seeing that how these self-amplifying RNA vaccines are working and what are the advantages and how they have been made, we are going to discuss in this session. So let us see the discussion with reference to the concept called as what are self-amplifying RNA vaccines. So in this, I hope you are following, my dear children. So self-amplifying RNA vaccines. So the approval of yet another RNA-based vaccine. It is based on RNA platform. An RNA-based vaccine for COVID-19 might not seem momentous, okay? But the endorsement last week by Japanese authorities of a job here means vaccine against SARS-CoV-2 constructed using a form of RNA that can make copies of itself inside cells. The first self-amplifying RNA, saRNA, granted full regulatory approval anywhere in the world. So this is the first self-amplifying RNA vaccine that has been given approval. Okay, so we can say this is the first saRNA vaccine that has been given approval and for a particular disease called as coronavirus or COVID-19. So the new vaccine platform could provide potent defense against various infections, and it can also be used to treat cancers. That is a momentous change, particularly with reference to this self-amplifying RNA vaccine. And because it could be used at lower doses, two advantages. One is this vaccine can be used to kill or destroy or eradicate the cancer. And the second one is this vaccine can also be used in the very, very lower doses of. It might have fewer side effects than other messenger RNA vaccines. So this vaccine, when compared to the other mRNA vaccines, this vaccine has very, very limited number of side effects, and it is needed in very small volume. And number three, this vaccine can be used even to treat cancers also. That is why this vaccine has a tremendous application. So this is the particular self-amplifying RNA vaccine diagram. So in this diagram, you can see small number of RNA molecules will be amplified into multiple numbers, and that amplification, how this is going to this small volume of RNA is going to be amplified, will be discussing. The newly authorized vaccine ARCT-154, you should remember this name now. The newly authorized vaccine ARCT-154, developed by Arcturus Therapeutics in San Diego, California, and its partner CSL, a biotechnology firm headquartered in Melbourne, Australia, triggered higher levels of virus-fighting antibodies that circulated the body for longer than did a standard mRNA vaccine. That is what. So it can able to stimulate more number of antibodies than the regular mRNA vaccine, and it can trigger it is needed in very lower number of antigens, or the pathogens are very small number of RNA molecules are needed. A company that develops saRNA vaccines. So they anticipate that with continuing advancements, saRNA technology will increasingly replace conventional mRNA. So just imagine how fast the technology is progressing. So, for example, earlier we can say till the last week or till the November, people thought that mRNA vaccine has a tremendous advantages, and mRNA vaccine is the ultimate vaccine. But now you can see one more vaccine that can able to be replaced by this vaccine is nothing but saRNA vaccine. Okay, so after the advent of saRNA vaccine, mRNA vaccine has become a very conventional vaccine. It has more versatility in its potential. Diversity of this vaccine and versatility of this vaccine has proved this vaccine so much powerful. Conventional mRNA-based COVID-19 shots consist mainly of the genetic instructions for viral protein that are surrounded by regular sequences. So you just go to the portal and watch my video on special vaccine called as mRNA vaccine. A cell's machinery produces the protein for as long as these instructions persist, and that protein, known as an antigen, stimulates an immune response. So actually, cells, how this mRNA vaccine produces, one mRNA copy of the targeted antibody sequence will be inserted, and that mRNA will go and stimulate the antigens and will act like an antigen, and this mRNA will with this stimulation, the antibodies are produced. As the antibodies are produced, and that they will be having the dynamic applications. Next, but whereas in the case of saRNA jabs go a step further by integrating genes needed for the replication and synthesis of the antigen encoding. So actually mRNA vaccines just carry an mRNA itself, and this mRNA will go to the ribosomal or protein synthesis machinery, and there at the protein synthesis machinery they encode and form the proteins which are responsible, which will act like that of the antibodies. But whereas in the case of saRNA will carry, self-amplifying RNA vaccines will carry certain genes, those genes which are exclusively meant for the replication purpose. So as it is having certain genes that are meant for the replication process, and this encoding RNA effectively establishing a biological printing process for fabricating the vaccine inside the cell. So this is a very important thing. We are not only sending an RNA, and we are sending an RNA. Along with the RNA, what we are sending one more important copy that can able to make this mRNA to replicate. Make this RNA to replicate. Once the RNA get replicated, we can see the dynamic changes. So because of which the RNA will be amplified like anything. So as the RNA has been amplified at the protein synthesis machinery of ribosomes, the RNA will be produced in multiple numbers. These RNAs which are produced in multiple numbers will be will able to provide protection against the pathogens. That is the very important thing. So what are the three distinct features? Number one, these vaccines are self-amplifying vaccines. Number two, we need very small volume of RNA encoded with replication gene. Number three, they produce enormous amount of proteins. Number four, as they are producing enormous amount of proteins, the fourth and last one is nothing but this saRNA vaccine has very limited and very less number of side effects when compared to the mRNA vaccine. As of now, mRNA vaccine is the only vaccine that has very limited number of side effects. But when compared to the mRNA vaccine, this vaccine is having very less number of side effects. So in this video, in this slide, you can see the images. Okay, so how the conventional mRNA vaccine is made and how the self-amplifying RNA vaccine is made. So here, a small RNA, from the small RNA, a antigen coding sequence will be taken. So try to remember now. In mRNA vaccine, antigen coding sequence has been integrated. Antigen coding sequence has been integrated. Whereas in the case of self-amplifying RNA, along RNA vaccine, so you can see here in red color, this is the antigen coding sequence. Okay, but along with this, there is a replicating gene also has been inserted, and this replicating gene will produce the more number of RNAs inside the host cell. or host body. And so, for example, in conventional mRNA vaccine, so if RNA delivered into the cell, and this RNA, viral antigens are produced, and these viral antigens ingested by cell that displays it to the body's immune system, that displays it to the body's immune system. But whereas in the case of, if you look at this, so yellow color, in this yellow color, you can say this is the replicating gene. Red color gene is the gene which is an antigen encoding gene. And once this encoding antigen encoding gene as well as replicating gene both have been entered, and they amplified copies of antigen encoding gene are produced. So these are viral antigens, and here you can see viral antigens. So where you can see more number of viral antigens, absolutely saRNA. Self-amplifying RNA vaccine has more number of viral antigens when compared to the regular mRNA vaccine. So that is how. What main clear-cut demarcation, clear-cut distinction is nothing but self-amplifying RNA vaccines are added with the replicating genes. Vaccine strategies compared. Presented with an RNA vaccine, the body cells produce antigens, protein sequences normally encoded by a virus, from a set of RNA instructions to prompt an immune response. If these instructions include the recipe for a replicase enzyme, that enzyme once created can make more copies. So which genes have been inserted are nothing but the genes that can able to produce a protein of an enzyme called as self-replicating enzyme. What is the self-replicating enzyme is nothing but replicase. This might mean that a vaccine made using this strategy can be given in smaller doses and elicit similar responses to conventional RNA vaccines. So when compared to the conventional RNA vaccine is nothing but mRNA vaccine. When compared to the mRNA vaccine, these self-amplifying RNA vaccines are needed in very, very smaller volumes. In the case of ARCT154, the antigen is a surface protein called spike that is expressed by SARS-CoV-2. Here also, antigen encoding gene has been inserted. Even in mRNA vaccine also, antigen encoding gene has been taken. The replication machinery is taken from a naturally occurring virus, a mosquito-borne pathogen known as Venezuelan equine encephalitis virus that causes deadly brain swelling in horses and humans. So actually, for making this, actually they cannot able to directly take the virus. For getting the virus, they had taken for getting that particular specific code of genes, they have chosen a virus named as Venezuelan equine encephalitis virus. And this Venezuelan equine encephalitis virus is a virus which causes brain swelling in horses and humans. Notably, scientists at Arcturus have removed key genes from the viral sequence backbone, thus rendering the key system non-infectious and safe for use in humans. Because of its virus-like nature, saRNA interacts with the immune system in distinctive ways that could prove beneficial across a range of disease scenarios. When it comes to preventing infections, for instance, its self-amplifying capabilities could enable the use of lower vaccine doses. When it comes to preventing infections, okay, for instance, its self-amplifying capabilities could enable the use of lower vaccine doses. ARCT154 requires 1/10 to 1/6 as much vaccine per person as other RNA-based vaccines are needed. For example, 10 molecules of RNA molecules are needed, but genes are needed in mRNA vaccine, whereas in the case of, particularly in saRNA, 6 molecules of RNA, 6 genes are needed. Reducing the amount of vaccine administered in each injection should result in lower production costs. So as the smaller size, a smaller number of viruses, a smaller number of RNA molecules we are giving, so thereby the cost of the virus and the cost of the vaccine is being cut down. Production cost reduces because of which the cost of the vaccine will also be reduced. And although the side effect of profile of ARCT154 seems comparable to that of a conventional mRNA shot, it is conceivable that the benefits of the platform's smaller doses will help to mitigate the severity of pains, fever, chills, and other loathsome symptoms collectively known as reactogenicity. So, as you are giving more volume of vaccine, the number of side effects could be more. But as the in saRNA, the vaccine that is needed to the body is very less. That is by absolutely side effects will also be very less. What we can say, less luggage, more comfort, more luggage, more side effects, less luggage, less comfort, less luggage, less side effects. So less luggage, more comfort. We are giving very, actually, when compared to the other vaccines and mRNA vaccine is carrying very smaller volume of genetic information. Even when compared to the mRNA vaccine, this saRNA vaccine is carrying very, very tiny volume of the genetic information. These unpleasant reactions remain a considerable impediment for people to take mRNA-based vaccines. Consider the seasonal influenza vaccine. Existing vaccines that use older vaccine technology cause only mild reactions. At present, several conventional mRNA-based flu jabs are progressing through clinical trials, and these are showing promising signs of eliciting more protective antibodies than existing shots. Yet their side effect profiles still leave room for improvement. And co-founder and chief scientific officer of Tiba Biotech in Cambridge, MIT, Massachusetts, the saRNA vaccines lower dose could help to solve some of the reactogenicity issues. What are reactogenicity issues? Once you are giving more number of pathogens, okay, as more volume of RNA, mRNA or DNA vaccine or antigen-based vaccine or live attenuated vaccine, whatever, if the vaccine, the pathogen enters, the antigen enters, and the reactions that are happening naturally in the human system can be collectively called as reactogenicity. The saRNA vaccine platform does have some downsides because of added genetic instructions. So one limitation, so don't think that it is the vaccine which is absolutely having only positive effects. Certainly not. It is having certain lower side effects. What is that? So we need to add certain genetic instructions. The jobs tend to contain longer sequences, typically at least 3 times the length of what is used in conventional mRNA. So remember, in mRNA vaccines, we are supposed to give very small mRNA piece. But whereas in the case of saRNA vaccine, we need to give more larger mRNA sequences, okay, larger RNA sequences. Why? Because self-amplifying replicase enzyme genes should also be inserted, which adds complexity to the manufacturing process. They also engage with the immune system in intricate ways, for example, by forming replication intermediates that help to stimulate beneficial immune signaling pathways. However, excessive stimulation can backfire, including when the vaccine prompts the immune system to block RNA replication, thereby nullifying its benefits. For example, excessive stimulation can backfire. For example, you are giving larger volume of this larger size of lengthy RNA you are giving. As you are trying to give lengthy RNA, that can sometimes it can backfire, including when the vaccine prompts the immune system to block RNA replication, thereby nullifying its benefits. More than a dozen saRNA vaccine candidates are currently in clinical trials for a range of applications, from shots for shingles and the flu to therapeutic vaccine for cancer, but researchers are already considering the platform's broader applications. There are some inherent advantages of saRNA if we are smart enough to take advantages from them. Actually, as of now, this is the only in under developmental phase. Once it comes to commercial aspect, it is going to have revolutionary changes, particularly in terms of vaccination. Even in terms of immunology, this vaccination has a tremendous positive impacts because small volume, low price. For example, why mRNA vaccine is not so much popular? Because that the cost of the mRNA vaccine is so high. But whereas in the case of when compared to the mRNA vaccines, saRNA vaccines are less cheaper, and because the volume of the saRNA vaccines that are supposed to give to the patients will be very less. So I hope you understand this. session. Thank you very much. We'll continue in another class.